Okay, here we are. This is lesson number 8 for SLs and HL students, the structure of DNA and RNA. So this is the topic 2.6, and we're almost at the end of topic 2, guys. So structure of DNA and RNA, here's your list from the IBO. The nucleic acids, DNA and RNA are polymers of nucleotides. DNA differs from RNA in the number of strands present, the base composition, and the type of pentose sugar. DNA is a double helix made of two antiparallel strands of nucleotides linked by hydrogen bonding between complementary base pairs. And Watson and Crick's elucidation of the structure of DNA using model making. I was going to do some drawing skills as well in this lesson. So here's our target for today. I can analyze and draw the structure of DNA and RNA. I can interpret the meaning of Watson and Crick's famous prediction in their article. So first things first. What is a nucleotide? All nucleic acids are made up of nucleotide bases. What are those bases for DNA and RNA? Well, hopefully you know that for DNA, they are adenine, thymine, guanine, cytosine, ATCG, and for RNA, we replace the thymine, the T, so the thymine with uracil. So the cytosine bases for each one. Okay, so DNA A, T, C, and G, and RNA A, U, C, and G. Now, what elements do we find within nucleotides? We did topic 2, carbohydrates, lipids, proteins. We talked about what elements are found in each one to identify them. So what are the ones in nucleotides? Look at our diagram here. We can see we have our sugar, which is just carbon, hydrogen, oxygen. We have some nitrogenous bases that has nitrogen, and we have a phosphate group here, phosphorus. So all the major organic elements are present in these. The sugar group and the phosphate group together make up the backbone of nucleic acid. So these two together are connected into a long chain to make up the backbone, either of single-stranded RNA or double-stranded DNA. And nitrogenous bases, they're what communicates the message. Okay, so take a look here at these diagrams. Take a minute to pause the video and compare and contrast them with each other. So if you're noticing here, the big difference is that we have a ribose sugar here, which has an oxygen atom here, and a deoxyribose here, which doesn't have an oxygen atom. Also, we're noticing that we have different bases here, so uracil here in RNA, but thymine in DNA. This is how the IBO wants you to draw DNA, is with circles for phosphates, pentagons for the sugars, and rectangles for the bases. So good diagram to have in your notes, guys. Okay, RNA versus DNA. So DNA has to use deoxyribose sugar for its backbone, while RNA can use ribose sugar. This is a must do. Do you have any idea why that must be the case? Think about their structures here. Well, DNA is double-stranded and RNA is single-stranded. So in order for it to fit and be able to come in this pattern here, the double helix, that extra oxygen on the RNA actually makes the ribose slightly too large to fit in this helical shape. So we have to have just a ribose sugar for DNA. So a deoxyribose is the ribose minus an oxygen. Okay. Watson Crick showed this very famous quote in their article, their Nature article. It's just posted on Schoology. You should definitely give it a read. It's only one page, double-sided, so 2 pages long. And it's a really good article for you guys to read. And they said it is probably impossible to build the structure with a ribose sugar in place of a deoxyribose, as the extra oxygen atom would make too close a van der Waals contact. So if they had an extra oxygen, it would make the van der Waals bonds, and the kind of bond in molecules we haven't talked about yet. We won't talk about more detail than this in this course, but that particular bond type won't work if there's not enough space between the molecules. And that extra oxygen is enough to mess that up. Okay, so thinking back to polymers in topic 2.3, 2.4, DNA and RNA are described as polymers of nucleotides. Why is that the case? Pause the video, talk to a partner about this. Well, polymers are chains of multiple molecules of the same type, and polymers of nucleotides are the subunit is nucleotide. The nucleotides here we have RNA nucleotides with the ribose sugar, nucleotides with the deoxyribose, different bases, and together they make up a polymer, many subunits bound together. Now, who remembers our reactions from topic 2 earlier? What kind of reaction do we use to join nucleotides together? Anybody? Anybody? So it's going to be a condensation reaction. We're going to be producing water is how we're going to bond them together. This is an anabolic reaction, bringing them together. Okay. And the similarities and differences between DNA and RNA, what are those? So again, one big thing we haven't talked about and seen yet is the single-stranded versus double-stranded. We have the bases here are hydrogen bonded together in DNA because there's two of each base. We have the deoxyribose versus the ribose, and we have uracil where we have thymine for DNA. Can you think of any other polymers we already talked about in this course? There's all the big carbohydrates, any polysaccharides, those are definitely polymers. Starch, cellulose, glycogen. Think about large protein molecules. You can also think about lipids like fatty acids, phospholipids. They're all polymers, large molecules, and many repeating subunits. Okay, I want you to take a DNA model kit, and there's a couple at the back. In the back cupboard by the aprons, there's two kinds of model kits. You're going to build one strand of DNA and one strand of RNA. Use the diagram to help. So pause the video for a while, take 10-15 minutes, and make a DNA and an RNA strand. Once you've made your strands, you can look at the key ideas here, write those down, check for understanding. And once you have that done, you want to make a complete DNA. So for bonding to happen of DNA bases, DNA has to run what's called anti-parallel direction. So we look here, DNA is going from 3' to 5' on this side, and 5 to 3 on this side. So it's going in opposite directions, but they are parallel to each other, hence the name anti-parallel. This allows them to fit together properly, and we use the 3' and 5' as ways to designate the directions because left and right, up and down don't work when the molecule can move in all directions. So the 3' and 5' are based on which carbon molecule the sugar is bonded to. The sugar is bonded to which phosphate. So here the third carbon is bonded to the phosphate, and here the 5th carbon is bonded to the phosphate. That's how we keep track of the direction. So make sure the model you built does in fact demonstrate this. Now you can choose to have used the Kinnex model kit or the Bio model kit. I personally prefer these ones, but both of them are just fine. So go ahead and finish building, tweaking your model if you need to. Make sure they are anti-parallel and they have the right shape. Once you're finished with your models, the next thing is to draw both DNA and RNA. So one of the skills from IB is to draw a representation of both DNA and RNA using pentagons for your sugar, circles for the phosphates, rectangles for the bases. Remember that carbon and guanine, cytosine and guanine are connected with 3 hydrogen bonds, and adenine and thymine are connected with 2 hydrogen bonds, and those are represented by little dashed lines, just like we talked about water properties in 2.2. So pause the video, use these drawings to help you, and make a drawing of both DNA and RNA and ask your teacher for feedback on the quality of your drawing. You have to do this from memory. Next thing I want to talk about is how DNA was discovered over time. Now, HLs will go into more about this later on, but for everybody else, it's important that you understand that structure of DNA was not something that Watson and Crick discovered. The word from the IB is elucidated, and that's because they took a ton of evidence from many other scientists and put the pieces together. So if all of these scientists were involved in this process, so we have Griffith, who figured out that DNA, the transferring principle existed, something could transform and carry information in cells. Avery figured out that it was DNA was transferring principle and not proteins. Then Hershey and Chase confirmed that because people had faults with Avery's experimental design, didn't believe him. They did a really amazing experiment, which you should look up, Hershey and Chase, to figure out how DNA is the transferring principle. And then Pauling figured out that that the helical form does exist in nature in proteins. Shagraf figured out that A and T, C and G are the bases in all organisms, but they come in different ratios, proportions. Rosalind Franklin and her boss, Maurice Wilkins, her supervisor, she came up with the X-ray crystallography that shows DNA associated with X's in the middle, which means it's a double helix. And then Crick and Watson, they're the ones who put all this together and built a model to figure out that it is a double helix with sugar, phosphate backbone, and nucleotide bases. So here is the model that Watson and Crick built, and they would have had a tech in the university lab build this for them based on their calculation specifications. This is the X-ray crystallography image that we'll talk a lot more about with HL that Rosalind Franklin came up with, that she was able to elucidate, and then was taken from her and used to figure out that X is a double helix. You can see the X means double helix. So how do models help scientists understand molecular biology? Well, we can't see molecules, so it helps understand the interaction between them. We can use molecules to help us do that, and that's what Watson and Crick did. Okay, and here's the last key ideas of this lesson. Okay, next part of the lesson is going to be 2.7, DNA replication, transcription, and translation. We're not going to do translation right away, so just focus on replication and transcription. Hope this is helpful and you have some nice models to show. Please post them on school so I can see some pictures, guys. Take care.